Namaskaram. My name is S. Ashok Kumar. Welcome to my channel. This part I am going to explain about the Zwitter ion. Okay. First I am writing something. Identify what is this one. Immediately you can tell that is an amino acid, but you have to tell like L-alpha amino acid. That is a way of remembering. For example, I am telling like before going to start that Zwitter ion, every amino acid within the cell existing as a charged form. Because if you take an amino acid, that amino acid, every amino acid having the amino group, that is a positively charged amino group, negatively charged acid group. For example, if you take, in case of protein, we are calling histones are positively charged proteins. So, reason we know. The positive charge of the histone protein contributed by the positively charged amino acids. Okay, na? Same way that we can call negatively charged proteins also. Na? The negative charge of those proteins because of the negative charge of the amino acids. As a result, what you need to understand? Within the cell, amino acids would like to exist as a charge. Okay, for example, I am telling the charge of an amino acid depends on the pH of the solvent. I will make you understand. For example, this amino acid, if you place it in the acidic pH solvent, when you tell acidic pH means the solvent consists of more number of protons, H plus ion. In that condition, NH2 by taking one proton, hydrogen, it becomes NH3 plus. Okay. Remaining COH group remains the same way, H and R. This is a positively charged molecule. This positively charged molecule we are calling as a cation. Why? Because it moves towards the cathode in the electrical field. Same amino acid, instead of acidic pH solvent, you can place in the alkaline pH solvent, where H plus ions are less or OH minus ions more. So here H plus ions are less. In this condition, what happens? NH2 remains the same. COH by losing proton, it becomes COO minus. H and R. This is a negatively charged molecule. This is called as a anion. Why? Because it moves towards the anode. So what I said clear now? The charge of an amino acid depends on the pH of the solvent. Now, this is a one condition. That is another condition, basic condition. Third condition, acidic, basic, we cannot tell. It is a particular pH for a particular amino acid. At that pH, these two combinations, COH become a COO minus, NH2 become a NH3 plus, H and R. Now I am asking you, this molecule moves towards the anode or cathode? Definitely, this molecule neither moves to the cathode nor moves towards the anode. This molecule, that electrically neutral molecule, we are calling as a Zwitter ion. You can call it as dipolar ion or amphoteric ion. That particular pH where particular amino acid existing as a Zwitter ion, that is a designating as a Pi. That is isoelectric point or isoelectric pH, where particular amino acid existing as a Zwitter ion. So each amino acid having its own isoelectric point, but randomly, roughly, isoelectric point of the most of the amino acids is around 7. This isoelectric point and isoelectric pH is very important in the technique, protein purification technique called as a isoelectric focusing that you will study in the next level. Now this is a concept of Zwitter ion. After concept of Zwitter ion, now I am going to explain structure of proteins. Actually, proteins having the 4 levels of structures include primary, secondary, tertiary or quaternary. I will tell you why I call tertiary or quaternary. Okay. Before going to tell different levels of protein structure, first I will explain the concept. For example, you take, this is a eukaryotic cell. Okay, na? This is the nucleus, this is the endoplasmic reticulum. Consider this is a rough endoplasmic reticulum. As we know, RER is a site of protein synthesis or site of translation process. Once the protein is synthesized, the protein enter into the lumen of endoplasmic reticulum only. The synthesized protein never enters into the cytoplasm directly. Once that protein is synthesized and released into the lumen of endoplasmic reticulum, the resulting structure is called as a primary structure. Once the primary structure is formed as a result of its synthesis, primary structure must convert into secondary. Secondary structure must convert into tertiary structure. This conversion, primary into tertiary structure, that conversion is called as a protein folding. Why that protein folding, you know? Tertiary structure is the most stable form of protein structure. When I said itself, you can understand primary structure is the least stable. Otherwise, tertiary structure is functional form of protein. When I said tertiary structure is the functional form, primary and secondary structures are not functional form. That is why once the primary structure formed as a result of its synthesis, primary structure must convert into tertiary structure to obtain the stability or to obtain the functionality. This concept you need to remember. Now I am going to explain different levels of protein structure. Now I am going to start with the primary structure of the protein. First I will define what is the primary structure of protein, then I will explain its importance. Primary structure of protein is, it is a linear sequence of amino acid residues linked by peptide bond. You should remember the definition. What is the primary structure of the proteins? You know, primary structure is the linear sequence of amino acids linked or stabilized by the peptide bond. You should remember the peptide bond. Here I use amino acid residues, that I will also explain. For example, if I take NH2, C, COOH, H and R1. Okay, now what is R1 means? Amino acid 1. This is which amino acid? You should remember this is L-alpha amino acid 1. Already I explained the structure of amino acid. One more I am taking here, NH2, CCOOH, HR2. What is this one? This is a L-alpha amino acid 2. Now, these two amino acids. How the peptide bond form, you know? The carboxyl group of first amino acid react with the amino group of the second amino acid. So, here OH, even a H. That results release of the water. Releasing of water we are calling as a dehydration. Wait for the point. Now I am writing NH2 CHR1C. Here this is a CO. Here the remaining NH. Correct na? Then CHR2 COOH. What you are seeing, this CONH is CONH bond is called as a peptide bond. Okay na? Now, I will explain point by point. First, I will explain, one more time I will explain about the formation of the peptide bond. Keen listen. This peptide bond is formed by carboxyl group of the first amino acid. react with the amino group of the second amino acid to form the peptide bond with the release of water. So, definition of the peptide bond you should remember. So, second point here it is that if I ask where this in a cell, where this peptide bond formation takes place, immediately you can call at the ribosomes, because ribosomes are the sites of protein synthesis. Here I can ask in two ways. In a cell where this peptide bond formation takes place, or in a cell where the protein synthesis takes place. For both questions, only one answer, that is at the ribosomes. Second point here it is that this peptide bond formation is mediated by a particular enzyme. That enzyme name is a peptidyl transferase. Very important enzyme. This enzyme concepts again I will tell you this one. What is the function of peptidyl transferase? Peptidyl transferase is an enzyme mediate formation of peptide bond during protein synthesis. Point is like when the peptide bond formed, there is a release of water. Yes or no? Here I can call it as a this is a one amino acid second. So this is a dipeptide. What is the definition of the dipeptide? Two amino acids linked by peptide bond. That is wrong. Two amino acid residues linked by one peptide bond. Why? Because peptide bond formation is an example of dehydration. Means a product formed as a result of dehydration, that product is called as a residue. Important point. A product is formed as a result of dehydration is called as a residue. That is why when you are defining a protein is a polymer of amino acid residues linked or stabilized by the peptide bond. Here, okay, next point is, here I showed the CONH bond is a peptide bond. Thing is that the bond between the carbon and nitrogen look like a single bond now. Exactly, that is not a single bond. Here between C and O there is a double bond is there now. One pi bond will shift here. Automatically here it will get the partial double bond. That is why peptide bond is having the partial double bond nature. Till now you understood now. Now this is a dipeptide. One more amino acid I will add and I will show you. Look here. This is a NH2, CCOOH, H and R3. Okay na? Here OH, here H. Removing as a water. Now this is a peptide bond, what you know. This is we can call as a tripeptide. What is the definition of tripeptide? Three amino acid residues linked by two peptide bonds. Like that you can add, for example, here I showed 3 amino acid added. Like you can add 9 amino acid. 9 amino acid, that is called as a nonapeptide. If it is a 51 amino acid residues added, that is called as a polypeptide chain. If it is a 1000 amino acid residues added, that is also called as a polypeptide chain. How much polypeptide chain you take? Last very important point I am telling with reference to the primary structure of protein. If you take the protein, you can see this is the first amino acid. First amino acid is also called as a left end. This is a last amino acid consider. This is called as a right end. With respect of the protein, left end, that is the first amino acid, which functional group is a free? NH2 group, because this is the carboxyl group engaged with the peptide bond. With the reference of the right end means the last amino acid, which functional group is a free? COOH. That is why I am telling, with reference of the protein, amino terminal or N terminal is at the left end. C terminal or carboxy terminal is at the right end. So, what you need to remember here it is the primary structure mean. Primary structure is the linear sequence of amino acid residues linked by peptide bond. One more point here you need to remember, last point with reference of the primary structure of protein. Here I showed the tripeptide. In tripeptide, how many peptide bonds are there? 2 peptide bonds. That is why, for example, how many number of peptide bonds present in a protein made up of 51 number of amino acid residues means you have to tell 50. That is when number of peptide bonds in a protein, that is equals to n minus 1. What is n? N is the number of amino acid residues. So this is about the primary structure of protein. After primary structure of protein, now I am going to explain the secondary structure of protein. Actually, what is the definition of secondary structure? You know, secondary structure is intermediate folding of a polypeptide chain. A polypeptide chain stabilized by hydrogen bonds. So, secondary structure I am calling as it is the intermediate folding of a polypeptide chain stabilized by the hydrogen bond. Actually, this is secondary structure, 2 types. Okay. One is helix. Helix. Second one is a sheet. Actually, among these 2 types, first discovered one helix. That is why designation given is a alpha. Second discovered one is a next discovered one is a sheet. That is why designation is a beta. That is why you can call secondary structure is of 2 types mainly, alpha helix and beta sheet. Okay. For example, if you take over 100 protein molecules, if you take in a cell, out of 100, or 20% of the molecule shows the beta sheet. Remaining 80% of the protein molecule exhibit the alpha helix. Why? You know, these alpha helix and beta sheet both are stabilized by the hydrogen bonds only. But while the helix is forming, helix allows greater formation of H bond. That is why greater the bonds, greater the stability, greater the prominency. That is why what you need to remember, alpha helix is the most common, or alpha helix is the most dominated secondary structure. Now just I will represent what is this one. This alpha helix again 2 types. One is right-handed alpha helix. Next one is a left-handed alpha helix. What I said, both are belongs to alpha helix only. But I am telling left-handed alpha helix most dominated secondary structure outside the cell. That means where? In vitro conditions, in the laboratory conditions. Now I am taking other alternative. Right-handed alpha helix is the most common secondary structure within the cell. Under normal physiological conditions of cell, right-handed alpha helix is the most common secondary structure. Now this right-handed, left-handed means, now you can observe this is my left hand. For example, if I fold my hand like this, the folding of polypeptide chain always you have to start from the thumb. Then polypeptide chain you have to rotate in the finger folding direction of the hand. See this one? Finger folding direction like this now. This is a N terminal, this is a C terminal. This helix is folded in the direction of the left hand. That is why this is called as a left-handed helix. Now same way you can observe like this. This is a N terminal, this is a C terminal. This is a right-handed helix. Now this is a hydrogen bonds. Hydrogen bonds either intra-hydrogen bonds or inter-hydrogen. Common thing I wrote here, it is a H bond. Now you take the beta sheet. Beta sheet is also very peculiar structure, 2 types. One is a parallel beta structure, parallel beta sheet. Next one is a anti-parallel. What it mean, you know? For example, this is a one polypeptide chain, N terminal, C terminal. This is a one more polypeptide chain, N terminal, C terminal. Between these two hydrogen bonds will form. Now this type is called as a parallel beta sheet. For example, this is a one polypeptide chain, N terminal, C terminal. One more polypeptide chain, this is a N terminal, this is a C terminal. Hydrogen bonds. Means this type is called as a anti-parallel beta sheet. So these are the different types of the secondary structure. You should remember only one point. What is the secondary structure mostly means? It is intermediate folding of polypeptide chain stabilized by the hydrogen bonds. One point. Next one, right-handed alpha helix is the most common or most dominated secondary structure in the physiological conditions of the cell. The remaining levels, tertiary structure and quaternary structure, I will explain in the next video. Till now, if you understand the concept, please like and share to your friends. Subscribe my channel. If you have any doubts, comment below. Definitely I will respond to your questions. Thank you.